Okay, this is the first of several preps that I will do for lecture 29. It's quite a lot of material, and I think it's better if presented in smaller, more sort of digestible chunks. So we're going to be looking at proximal distal outgrowth and anterior posterior specification, that specification of the digits in this lecture prep. And I'll just point out that we're looking mostly at the chick forelimb, and from anterior to posterior, the digits are digits 2, 3, and 4. And the basic sort of vertebrate limb digits is 5, and they're numbered. There's a sort of standard 1 through 5. Your baby finger is digit 5, your thumb is digit 1, and we know, I guess, based on sort of developmental information and gene expression and so on, that in the chick embryo, the first of the 3 digits in the wing, the forelimb, is equivalent to number 2 in the vertebrate standard one. The second one is number 3, and the third one is number 4. So that's just standard numbering. Yeah, and what it means is that the first and the fifth don't develop in this particular limb. These are the learning outcomes, which I will go over in the class. There's a number of signals that we're going to go through over the course of all the prep material. And so the key for being able to describe the signals is, first of all, knowing which affects which, but also how they intersect, because there is some interaction between, as the limb is developing, dorsal versus ventral doesn't happen at a different time than the rest of it. And the anterior posterior is being specified as outgrowth proceeds, but it's strongest, of course, in the later stages where the most distal structures are developing and so on. And when we ask you to understand the genes, of course, we want both, what are the gene products doing? What are the proteins that are expressed from those genes doing? And then what happens if you knock it out? What happens if Sonic Hedgehog just isn't expressed in the developing limb bud? What's going to happen? And then misexpression, what if we express it in the anterior as well as the posterior, or instead of the posterior? And so be able to predict those kinds of outcomes. And we'll also describe the role of apoptosis in digit development. So as I've said a few times in class, the embryo develops according to a certain plan, a certain set of procedures and kind of processes. And one of them involves, in some cases, like in the nervous system development, and in other kinds of development as well, sometimes producing more cells than are needed. And then some of them go through programmed cell death, which is a very regimented kind of process that causes a very orderly killing off of the cells. So certain structures fall apart and are broken down, and the materials can be used in other cells, right? So nothing is wasted. Okay, and so limb bud patterning. So the limb bud, if you look at the kinds of experiments, you can think about what we talked about in a previous class, was the removal of the apical ectodermal ridge. If you take that out, you stop development, further outgrowth of the limb bud, and then whatever has taken place, that's what you see. So you see the proximal most structures, and then if you remove the apical ectodermal ridge a little later, you will see sort of proximal plus intermediate, and then if you wait till even later, you get almost the whole limb except maybe the very tips of the digits and so on. And there's other kinds of experiments that gave researchers the idea that, um, you know, cells appear to be acquiring information at a certain time, but then they interpret and act on it later. In this case, after leaving the progress zone. And if you think back to somites, there's a direct analogy with the somites, because the somites too acquire information at an earlier stage, and then they form according to a clock um, that is autonomous. They're not affected by the other cells around them, and they form the type of anterior versus posterior somite that they're specified to produce, even if you move them to a different location, right? So this is not a new idea, that cells acquire some positional information, which means here's where you are in the body, anterior versus posterior, dorsal versus ventral, etc. And so this is the structure you're supposed to be contributing to. And it's not until later, in this case, after leaving the progress zone. Remember, that's the undifferentiated cells right below the apical ectodermal ridge. That's where cell proliferation is occurring. And some of those cells are stem cells, and some of those cells are cells that are going to form differentiated structures after they, quote unquote, leave the progress zone. Instead of leaving the progress zone, it's more as if the progress zone kind of moves along and leaves them behind. I think of it as like a motorboat on the surface of the water, and it's leaving the wake behind, the ruffles in the water. And those ruffles are the cells that are left behind. And another piece of information that we're going to be looking at is, that there are signals coming from different parts of the limb bud that give the cells information on proximal versus distal, anterior versus posterior, and dorsal versus ventral. But as I said before, you will see that they kind of, some of them interact with each other, which we won't see in this first one. So I want to start with proximal distal specification and patterning. And so there are 3 different models, and the first 2 each explained some of the data and some of the kinds of experimental results that people were getting from different kinds of manipulations, but neither one seemed to explain all of it. And if you're trying to figure out how something works, you can't have experiments that contradict each other or give you different results. You can have that, but what that means is that the model or the hypothesis you're working with is not correct, right? It's not we want the most correct information, the most correct model we can have, and that has to be one that explains all of our results. And so the first one is called the progress zone model. So remember, the apical ectodermal ridge sits, it's a thickened group of ectodermal cells, sits on the distal tip of the limb bud. And then the progress zone is where the cells are dividing. And that division contributes a lot to the outgrowth. But remember that, especially in the early outgrowth stages, there's also migration that's happening. But that migration is probably not sort of right in this area of rapid division. And so the idea is you've got cells in here. Now, these cells are dividing, so it's not that each cell is experiencing a certain amount of cell division, but that it's experiencing it and its daughter cells. Maybe there's a way of counting the numbers of cell divisions. Maybe it's just a matter of signals that they're receiving here. So the longer you spend in this zone, the more of the signals that you've been receiving. So maybe if you have a short blast of a signal and you get it, for just a few minutes, for just one time, that gives you one set of instructions. If you have the signal over a few hours, you have another set of gene expressions that come up. And then if you have like 12 hours of it or something like that, then you have different fate as well. So there's different ways of thinking about this, but the way if you were going to tie it down, it would basically be the more time that is spent in the progress zone, the more distal the structures. I hope you can't hear that. It's after hours, so there's no reason why there should be all this thumping and smashing. Cells that spend very little time in that progress zone, and then the progress zone moves along and kind of leaves them behind, these are going to form the most proximal structures. So think of the upper arm, right, the humerus. And then if a little more time is spent in that progress zone before the cell is left behind, then we're going to have more intermediate structures like the radius and ulna. And then the ones that have spent the longest in the progress zone before being left behind, those are going to end up forming the wrists and the digits, right? So that's the idea. It's just how long did you spend? And if we know how long you spent on this axis, we can extrapolate and go, okay, for this many hours, then you're going to be forming the tail end, the most distal part of the humerus, and the beginning part of the radius and ulna, or something like that. So the idea is that just based on time spent in the zone, you can then extrapolate and figure out exactly what that batch of cells is going to form. The second model, I think, arose probably most likely because it was already known that there was this FGF signaling that was taking place. If you remember, you had FGF10 in the cells immediately below the ridge, causing FGF8 to be expressed in the ridge, and then that signaled back to the cells underneath to to express FGF10 again. So because it was known that an FGF signal was emanating from this region, the idea was, what if what we're seeing is these two different gradients? And we've already talked about some kinds of patterning that occur because of two antagonistic gradients. And so this is not sort of particularly unusual. And the idea is that you have some sort of signal that emanates from the proximal region and another from the distal region. And if you want an analogy, think about the somites, newly formed somites, and the node in somite formation. And this is an example of two opposing gradients that lead to the definition of where each somite boundary is forming. So a lot of the elements of this model kind of make sense, and because it's been seen in somite formation, which is almost finished, I think by the time, well, no, the most anterior ones are finished by the time the limb bud is forming. And so, that's the idea. And so, you have a gradient that the source is the most proximal region, and then there's diffusion away as you go towards the distal, and then distal apical ectodermal ridge is the source of a signal that diffuses away. And so as development proceeds, cells that are sort of in the high level of the first gradient and with little or no expression of the more distal one are going to form your proximal structures. The cells that are mostly in the distal side that have most of this signaling molecule but very little of the other are going to form your more distal structures. And then the ones in the middle that are getting equal or relatively similar amounts of each of those two gradients, there's enough of it that it's considered to be overlapping. There's meaningful signal from both sides are going to form your intermediate structures. So that's the idea. It's basically just two sets of gradients, and they overlap. And the different relative amounts of each of the molecules specifies fates along that continuum. And so this is interesting because the molecules that we're going to just quickly talk about are ones you've already seen with somite formation. So it is known that retinoic acid is formed in the proximal region of the limb bud, and then there's a graded expression. So that's what this orange line is, or red, I guess that is. And we already know that FGF is produced from the apical ectodermal ridge, and that that's going to produce it. These are both diffusible signals, and so we know that there are going to be these opposing gradients. So the idea is, if you're in the red zone, you're going to form proximal structures, and the more of the retinoic acid signal you're receiving, the more proximal structures you're going to form. And the less you have, then the more towards the intermediate structures. On the distal side, the more FGF you have, the more distal the fates, until we get to this region sort of in the middle where you have, again, meaningful amounts of both signals. You're going to get the more intermediate structures. Okay, and then so both of these models made sense, and there was data that supported, but there wasn't data that exclusively always. There was no one model that all the data supported, I guess is the better way to put it. I think I was going backwards there, and so this is the current model that they're working with. And it suggests that there, in the proximal and the distal and intermediate areas, there are different ways that this is actually functioning. So if you just look sort of in the early limb bud, we still have this retinoic acid signal. And this retinoic acid signal is opposed by FGF from the apical ectodermal ridge and a protein called CYP26B1. So it's just it inhibits the signaling, and so the repression is coming all the way along from the distal side, and that confines the retinoic acid gradient just to the most proximal region. So it doesn't extend all the way across. One of the pieces of evidence was simply that you couldn't document sort of this nice graded distribution, which is what you might expect from the previous two-signal model. And so the idea was the proximal region has high levels of retinoic acid, and the more retinoic acid you have, just like in the previous one, the more proximal your structure is. And so in the just the proximal part of the limb bud, you can form the humerus or part of the humerus based on levels of retinoic acid. Once the retinoic acid, though, drops below a certain threshold amount where it's not really meaningful, there's not enough to activate any major gene expression, then what we're doing is we're looking at the time in the progress zone based on probably FGF signaling and or numbers of cell divisions and that sort of thing. There's a number of different waves of gene expression. There's multiple things that could be happening in this progress zone, but basically the time spent is now directly connected to which types of cells you're going to form. So this part is specified by the amount of retinoic acid, but everything else over here is based on time spent in the progress zone. Okay. So the currently accepted model is the third one, and there is still some question about whether retinoic acid is really the signal, but there is some evidence for it. So if you lose activity of this gene, And this gene encodes a protein that basically kind of binds to and metabolizes, breaks down the retinoic acid, and is very important for the gradient formation. And so if you lose this, then the limbs are truncated. That means they are shorter than normal, and they're missing some structures. And we know that the target genes of retinoic acid, which is it's a signal that activates, of course, gene expression. Um, the proteins, the homeodomain proteins that are targeted, are known to have an effect on proximal-distal patterning. So this isn't 100% certain, but it certainly is strong evidence. And then if you look in the distal limb bud, you have oscillations of gene expression, and this is similar to the oscillations that were discussed very briefly when we're talking about somite formation. And Harry2, I don't think I mentioned this specifically in the lecture, but Harry2 is known to have oscillations of gene expression in the distal limb bud in that proximal zone. And whenever you see genes that are being turned on and then repressed, and then turned on again and repressed, and turned on and repressed again, this suggests some kind of counting mechanism that is related to then timing. And so the more cycles of this Harry2 expression and or lots of other genes that are probably oscillating as well, then the more distal you are. And there is a connection here, even though I didn't really talk about it, because there's lots of genes now, more than just the hairy gene, which is a Wnt target, I think, if I remember correctly. Yeah, so there's many of these oscillating in somite formation, and probably the same is true in limb bud. So this is the evidence. We know the sort of the retinoic acid gradient evidence, and we know the oscillation part. There is additional evidence, of course. These are the kind of the compelling ones that that make it seem like this is the correct model. Okay, and I'm going to stop there, and I will start up again for the next section, because I just don't want these to get too long.